Okay, take it away.
Thank you, Donna. Hi, everyone. This is Clara Wenjing. I'm from the Jeffries Lab. And today I'm going to talk about a type 2 innate lymphoid cells, ILC2s, in the tumor setting. Next slide, please. So the concept of using host immune system to defend cancer has been an ongoing focus of cancer research for more than a century. And in the past decade, immunotherapy is becoming a new emerging pillar for cancer treatment, along with chemotherapy, radiation, surgery, and targeted therapy. Popularity of immunotherapy is evident through the growing number of active drugs that are being developed over the years. Among the different types of immunotherapy, cell therapy has taken the center stage, possibly due to the successful cases such as the CAR T therapy, TIL therapy, dendritic cell-based therapy, many of which are approved by FDA. Cell-based therapy definitely presents a great potentials in treating cancers, and there's a great need for exploring other immune cells that can be put into this use. Next slide, please. One more click. Yeah, thanks. One of those cell types that can be further explored is innate lymphoid cell type 2. ILC2s are a group of innate effectors that mediate type 2 immunity by driving the defensive response against helminth infection and sensitizing the host to allergens that drives allergy and lung inflammation. In cancer setting, the presence and activation of ILC2s can have pro-tumorigenic effect. Some of the mechanisms include the recruitment of MDSCs and M2 macrophages by IL-13 released by ILC2s, and those are cell types that are well known for their immunosuppressive roles. Also, IL-13 has the ability to remodel and maintain lung tissues during infection. Overdoing this may lead to fibrosis. On the other hand, ILC2s can also have anti-tumorigenic effects. IL-5, the cytokine, can activate eosinophils, which release potent granules that are effective at eliminating neoplastic tumor cells. It can also mount NK and CD8 T cells, which are the keys in anti-tumorigenic processes. Our lab has also found that overexpression of IL-33 can prevent metastasis in lung and prostate cancer. In fact, this is how we started to study ILC2s because we found that metastatic cell lines that have lost the expression of IL-33 and IL-33 being the potent activator for ILC2s, it naturally draws us to this immune cell type. So conventionally, ILC2s are believed to be a homogeneous cell population with a type 2 immunity. However, more recent studies reveal that there is a plasticity in this cell population. By changing transcriptional profiles, the cells are able to adapt to different circumstances in a flexible manner. And this has been shown to have implications in cancer setting that can be potentially be harnessed for therapy. Next slide, please. So to start off, let's look at the effect of ILC2s from tumor-bearing animals. This is done by our postdoc, Dr. Irina Sarachonen from Jeffries Lab, where ILC2s were isolated from tumor-bearing donors, cultured for 5 days with the presence of activators, and then put into recipient mice which were transplanted with tumor cells. The result shows that there is a significant decrease in tumor volume and reduced disease severity in the ILC2 treated animals. And figure C and D are gymostin stain of eosinophils on tumor cross-sections. C is the control and D is the treated. And as you can see here, adoptively transferred ILC2s allow eosinophils to infiltrate into the tumor tissue, facilitating the anti-tumorigenic effect by eosinophils through releasing of the granules. And this is likely the result of IL-15 production by ILC2s that facilitated the migration and infiltration of eosinophils. And next slide, please. So we wanted to know what happens when to the ILC2 population during tumor development. So we did a single cell sequencing of the ILC2 isolated from the lungs of tumor-bearing mice. Kind of surprisingly, we see a great heterogeneity in this supposedly homogeneous population based on the transcriptomic profiles. I won't go into details of each subtype, but there's definitely a couple of really interesting ones here. So for example, cluster 0 and 1 have upregulated markers that are important for T cell recruitment, as well as multiple interferon induced genes, which indicating that this is the Th1 signature. We also have antigen presentation pathway enriched subtypes. They express some of the key antigen presentation genes. And this suggests that in the tumor setting, ILC2s can be equipped with antigen presentation abilities and may be able to instigate interactions with antigen-specific T cells and facilitate adaptive immunity. Also, we have cluster 9, for example, that express significantly higher level of Th1 genes such as granzyme B, granzyme C, and FITM1. So definitely an interesting subtype that we see there's a shift from Th2 subtype, Th2 phenotype to a Th1. The take-home message from this slide is that the LC2s is a heterogeneous population that is equipped with different transcriptional profiles modified in response to environmental changes created by the growing tumor. And this heterogeneity may define the LC2 functional plasticity promoted by growing tumor microenvironment. Next slide, please. So the last point, to compare how growing tumor naturally affect the LC2 expression and how the ex vivo stimulation can possibly modify cells for immunotherapy, we conducted a comparative analysis of non-activated versus activated LC2s. Activation is through co-culturing of LC2s with their activators for 7 days and then sent for sequencing. And upon activation, we see that T cell recruitment markers, Th1 immunity markers, and antigen presentation markers increase in the total population. And this implies that ex vivo activation of LC2s may enable them to acquire associated anti-tumorigenic effector functions. So collectively, we identified a heterogeneous LC2 population with different immune subtypes that can be shifted towards a more pro-inflammatory genotype from upon activation, and with the main contribution to T cell recruitment power. This shift suggests that ex vivo manipulations can modify transcriptional profiles of LC2 and better prepare them for cell-based immunotherapy. And even better, maybe we can select for subtypes that are most effective at eliminating tumors. And thank you. And I'll be happy to answer questions.
Great. Thank you very much, Clara. So questions? Dr. Nee, do you have a question?
Oh, yes. So very nice work, Clara. So yeah, regarding this ILC2 cell population, as you say, that's a pretty heterogeneity. So so how many type of tumor cell model you started?
So this study kind of started from 2 of the tumor types that we studied. One is prostate, one is lung. And through those paired cell lines, primary versus metastasis cell lines, we found there is a drop in IL-33 levels. And then IL-33 being the potent activator for LC2, so we started looking into this immune cell population. And part of my thesis is to hopefully establish the link between the tumor expression of IL-33 with the LC2s and along with all other areas of antigen presentation to make a broader scope of what's going on in the tumor microenvironment.
Mm-hmm. To my understanding, did you see this Th1 like immune response has been enhanced if you transfer this ILC2 cells to the recipient animals?
Yeah, so we definitely see a response when we transfer the LC2 into the recipient. So we see a lower tumor volumes and better survivability. I believe there is also data supporting that there are Th1 cytokines being produced in the microenvironment of the tumor setting. Although I didn't show it here, but on top of my head, I can remember there is definitely a cytokine profiling showing that there is a shift towards Th1 phenotype.
Thank you. Thank you. Great work.
Thank you.